Good morning dear students, welcome to Genesis. In this lecture I am mainly going to cover guttation, experiments which are related to conduction of water through plants and forces responsible for ascent of sap. Well, guttation is such a phenomena which is usually seen in plants which are short in height, okay, like say for example grass, tomato, then various kind of ferns, banana plant, all these are short in height. As a result of which in these plants sometimes the root pressure that develops, it becomes so high that they start to lose water from the margins of leaf vein. So in a leaf these are the veins, and from the margins of the leaf vein they start to lose water, and water in this case is lost in the form of water droplets, unlike in transpiration. In transpiration the water is lost through stomata which is present on the upper and lower surface of the leaf in the form of water vapor, but in case of guttation the water is lost from the margins of the leaf veins through pores which are called hydathodes. These are the structures. Like in plants, when you study transpiration we use the term stomata, here we use the term hydathodes. These are present right at the margin where the leaf veins come to end. And in this phenomena, remember one thing, water is lost in the form of water droplets. And why is this happening? This happens because of increased root pressure. I have already discussed that root pressure is that pressure which gets developed in the root cells due to cell osmosis of water. So guttation, transpiration, these are various ways by which a plant can lose water. And why did I mention that it is mostly seen in plants which are short in height? Because in plants which are short in height, the root pressure is enough to push the water to the topmost leaves of the plant, which may not be the case with tall plants. In tall plants, the root pressure definitely helps in pushing the water through the stem, but there are other forces which are also playing role in pulling the water to such great height. We will study that afterwards. So now let's go and see how the root hair cells, they absorb water and salt and minerals from the soil. As you already know that the root hair cells are nothing but extension of epidermal cells. So for example, if I draw epidermal cells one after another, then this is how you will see that the root hair develops from the epidermal cell. Now imagine this root hair to be surrounded by soil particles all around itself, and right around the soil particle there is envelope of water. This is how it is present. So there is envelope of water around the soil particles. This water is very important to the root hair because the plant requires this water. So there are two things that the root hair is going to absorb. Number one, just water, and number two, salts and minerals. Now there are two separate mechanisms that this cell will apply to absorb these things. Now for absorption of water, like I have already told you, the concentration of cell sap of a root hair cell, the concentration is all of what? Of the salts, concentration of salts, okay? The concentration of salts in the cell sap of the root hair cell is always more compared to the water in the soil. So can I indirectly say this, that inside the vacuole of the root hair cell, the concentration of water is less compared to that in the soil. As a result of which the root hair cell will absorb water from the cell by the process of osmosis. Basic osmosis. Why? There will be movement of water molecule from higher gradient to lower gradient across a semi-permeable membrane. So the process is osmosis here. But when we talk about salts and minerals, in my last class I explained it already, that the amount of salts and minerals which are present in the cell sap is already higher compared to that is present in the water. Yet this cell is going to absorb salts and minerals from the soil. How can it do? It has to absorb salts and minerals actually from a lower concentration gradient to a higher concentration gradient. So I'm sure you already know this answer. This will happen by the process of active transport. where there will be movement of solute from lower concentration gradient to higher concentration gradient across a semi-permeable membrane, and this cannot happen free of cost. So there will be utilization of ATP. So now very clearly you can say that a root hair absorbs water by the process of osmosis, where there is no usage of ATP, but to absorb salts and minerals it has to perform active transport and will have to shell out ATP. Now let us discuss certain experiments to understand conduction of water in a plant. So we will start with experiment number 1. There are total 4 experiments that we have to study. So in the first experiment we take a plant which is called balsam, and we take 2 test tubes. In both the test tubes we put equal amount of water. In test tube A you do not put any plant, but you just make sure that the level of water is covered by a layer of oil. The same is done here, that the level of water is again covered by a layer of oil. So here is water and the level of water is covered by a layer of oil. Why are we doing this? Because we want to ensure that there is no loss of water by the process of evaporation. So this is A and this is B, but in B what we do is we take a proper plant, as in we uproot the plant of balsam and we put it inside this. Why are we doing this? Because we want to see that the roots and the plant when kept in water will absorb water. We want to see that happening. So now you keep this setup as it is for a couple of days, and after some days you will find that there is no change in this setup, but the setup B shows a decrease in the level of the water. And since we have already made sure that there was no loss of water by evaporation, then 100% this loss of water has happened because the roots of the plant must have absorbed water. That is why you see a decrease in the level of water. So this was experiment 1. The specimen used is balsam, and by this method we are trying to show that there is absorption of water by the roots of a plant. My question to you is, what is the reason of keeping A in this experiment? If you can tell me what is A in this experiment, write down in the comment box. In experiment number 2, the specimen is again balsam, and in this what we do is we take a beaker and we fill it with water, but we make sure that the water is mixed with a stain called eosin stain. It's a very common pink in color. So what will happen, this entire water in the beaker will turn pink, and now you put this rooted balsam tree in it along with its leaves, stem and roots, and you keep the experimental setup as it is for 3 to 4 hours. After 3 to 4 hours, we are going to take transverse section of number 1 leaf, stem and roots, and we will observe it under the microscope. Under microscope, you are going to see that every single part of the specimen that you are observing remains whitish in color, but one particular part takes the color of eosin. That part is the xylem. So from this experiment, what we can prove is the xylem is present in the root, it is present in the stem, and it goes up to the leaves. So basically the water which is absorbed by the root will pass through the xylem from the root to the stem to the leaves. Experimental 3 is about a process called girdling. You know what is girdling? Girdling is a process where you either take out the xylem of the plant keeping the phloem intact, or you take out phloem of the plant keeping the xylem intact. But to understand this, you must have this idea that if this is a part of the stem, I'm talking about stem, then in a stem it is always the xylem which is right in the middle and it is surrounded by phloem. So this part is xylem and the outer part is phloem. Functionally, you must know the difference that xylem basically is into conduction of water and phloem does transportation of the food that is prepared in the leaves. This is why the direction of movement of the substances that they conduct is also different. In case of xylem, the movement of water is always from the roots to higher parts of the plant. But in case of phloem, the transportation of the food is from the leaves to parts which are above it and also to parts which are below it. So it's a bidirectional movement in phloem, but in case of xylem, it is unidirectional movement of water. So now when we practice girdling, what we do is in some experiments, we are going to cut a portion of the stem in such a way that the phloem remains intact and the xylem is lost, and in another case, we will keep the xylem and the phloem is lost. The third experiment, dear students, we are going to use the process of girdling, but in this, the specimen that we have taken is balsam. That means we have taken small shoots of balsam and we have submerged them in beaker containing water. In the first case, what we have done is we have performed girdling and removed phloem, and in the second case, we have removed the xylem, keeping the phloem intact. Now, in both the cases, you will see here, the portion which is right above the cut has leaves attached to it, and these leaves are turgid, they are intact, okay? Whereas in the other case, just above the cut, the leaves have drooped. What could be the reason behind this? Here it is clearly understood that since the xylem is intact, the water has been absorbed and has been sent upwards to the leaves, because of which the leaves are turgid. Whereas in the second case, since the phloem is kept but there is no xylem, the transportation of water to the leaves got hindered. These leaves could not get water and they drooped. They are no more turgid. Now in experiment 4, we are not taking balsam, rather we are taking a rooted plant along with the pot. So here we take guava, or you can also take any tree, but we are going to take out the soft stem of that particular tree, okay? That means we don't want a tree, but a potted plant of the variety either guava or any other tree. But in this, if you look carefully, we are going to take out a small part of the stem from where we have removed the phloem, and we have removed the phloem keeping the xylem intact. Now we are going to observe this setup for few weeks, and after few weeks we will observe that the part which is right above the cut increases in diameter, and the part which is below the cut, it either becomes weak or dies out. Now what basically is happening here is, in the plants, due to the process of photosynthesis, food is manufactured. Now this food goes to parts which are higher above it as well as to the parts which are below it, that means to the root and to the lower half of the stem. Now here, since we have removed phloem, that passage of food is obstructed, as a result of which this part of the stem is swollen with the accumulation of food. The food will accumulate here, but because of the absence of phloem, it will not be passed down. So this clearly shows that in the first 3 experiments, we showed how xylem is very, very important for conduction of water. And in this last experiment, we are trying to show the importance of phloem in transportation of food. In experiment number 3 and in experiment number 4, we have also performed a special method which is known as girdling, where we basically remove either the xylem keeping phloem intact or vice versa. Now let's quickly see which are the forces that play a major role in ascent of sap. Ascent of sap. Ascent of sap simply means the upward movement of water and the dissolved salts and minerals with it from the lower part of the plant body to upper part of the plant body. Now this may sound simple, but it's a very complicated process. Imagine plants like coconut tree, mango tree, which are so tall, there you are transporting water against gravity. So otherwise, like when we look around ourselves in big apartments or buildings, to draw water to height, we usually use a pump, but we don't find such pumps in a plant. Then what is the mechanism because of which a plant can pull water up to such great height even against gravity? This is achieved because of certain forces which the plant develops within itself. Now the first force because of which this is possible is called the transpirational pull. What is transpirational pull? It is that suction force which is developed in the stem of a plant due to the loss of water from the stomata of the leaves. So there is a continuous loss of water which is happening in these plants, and because of this there is a suction pull developed within the stem of the plant, or in case of trees. Now remember one thing, this plays a major role especially in plants which are very tall, because there is another force which also plays similar role for ascent of sap, which is known as the root pressure. This we have already studied. This is basically the pressure which is developed within the plant due to cell osmosis of water. But this root pressure is not strong enough to force water to greater heights, like especially for coconut tree, for big tall trees, this itself may not be enough. So there, along with the root pressure, transpirational pull is that main power which helps in drawing water to such great height. The third force which helps in this process is known as capillarity. What is capillarity? It is that property by which water or any other fluid can be drawn upwards to greater height only if the diameter of that conducting pipe is less. What I try to mean is, if you are using a thin pipe to draw water in comparison to a pipe which is thicker, then in the pipe which is thin, drawing water is going to be easier compared to the other one. So capillarity is seen in mostly those plants which are very tall, like for example coconut plant. If you observe carefully, the trunk of the coconut plant is way thinner compared to any other plant which we see around. Like for example a mango tree. A mango tree will have a thicker trunk compared to coconut tree. Now what is here I have drawn two trunks. Water can easily rise up through this because it has smaller diameter comparison to this because its diameter is way thicker than this one. This is capillarity, which you see in plants which are very tall. So if a plant is very tall, we can expect its stem to be little thinner in diameter because that will help in the process of ascent of sap. The fourth property that helps in this is called adhesion. What is adhesion? Adhesion is the property by which the water molecules can remain attached to the surface of the xylem while they are transported upwards. This helps in a regulated movement of water through the stem. So there is attraction between the water molecule and the cells of the plant. This is helping in pulling them upwards. There is another force also which is known as cohesion. Cohesion is that force which is between two water molecules. That is equally important for this ascent of sap. So when there is a force of attraction between two different molecules, we call it adhesion, and if it is between two similar molecules, like between two water molecules, we call it cohesion. So for ascent of sap to happen, there are four major forces that are playing. Number one is transpirational pull, number two root pressure, and like I have told, root pressure is enough to draw water in smaller plants like tomato, like grass, but it may not be enough for plants which are very tall. So in such case, along with root pressure, the suction pull is generated by the process of transpiration. Then capillarity, this is that property because of which plants with thinner diameter or thinner trunk can easily draw water to greater heights. And fourth is adhesion. It is the force of attraction between the water molecule and the plant cells. Okay. With this, we finish this chapter. Do watch my shorts for a quick analysis of this chapter, and we are going to meet soon in my next video.